May or may not work out. I didn't bring my normal, the snowball microphone. I brought it, but I don't have the cable for it, so I'm gonna have to be kind of, maybe I'll be a little anchored here, which is not my optimal, but all right.
Good weekend? Yeah.
So this week is metabolism. In particular, well, next 2 weeks are metabolism. So we're going to talk about catabolism. And the next week we'll talk about anabolism. And basically the idea today is we're going to talk about how we break stuff down and make energy out of it. And then next week we'll talk about how do we build stuff back up from the energy. So we'll start off with a case study. And I'm going to make everyone answer. So, even if you say, well, I'll agree with him or her or whoever or you or whatever. 59-year-old woman with type 2 diabetes for 14 years. Presented with a history of 3 months of vague abdominal pain and 4 to 5 loose bowel movements daily. That sounds really not that much fun. Basically, that's diarrhea 5 times a day. That does not sound good. Which worsened over the 4 days before admission to the hospital. On the day of the admission, she reported weakness, dizziness, blurred vision. Her husband had noticed slurred speech and a reduced level of consciousness. She had documented normal renal function 4 months before this admission. Her diabetes was well controlled with metformin for the past 8 years, so she's obviously has some diabetic issues. 3 months before admission, she started Orlistat, and that coincided with the onset of abdominal pain and chronic diarrhea. Preliminary laboratory investigation showed a life-threatening metabolic acidosis with a pH of 6.5. She had bicarbonate of 2 millimoles per liter and a base excess of 38 millimolar per liter. Blood glucose was 5.6 and serum lactate was 23.4. A blood metformin level measured at 30 milligrams per liter, where therapeutic levels are less than 2 milligrams per liter. So, I'll give you a minute. Take a look. The key is, obviously this is drugs, so I'll give you a little hint. It has to do with drugs, the drug she's on. See if you can find something. And if you find something, you can just shout it out.
Is she on like way too much metformin?
Yes, that's the answer. And that is metformin-associated lactic acidosis. That's obviously a pretty simple diagnosis, I would think, based on... Go ahead.
So the answer you said was that she was on too much metformin, but they hadn't changed her metformin. And
That's
she
true.
was on a good dose of metformin for 8 years. So how come the dose of metformin that she was fine at for 8 years is now all of a sudden too much? What changed? So what does the word that do?
Google says it helps people lose weight.
Okay.
It stops the enzymes from becoming fat.
Okay.
So the metformin dosage was way too high for the amount of the weight that she had on her after she started taking it.
Okay. So maybe her weight significantly changed. The other part that you guys are looking at here is renal function was markedly impaired. So now we have an impaired renal function. What happens when we have an impaired renal function? How do we excrete metformin? Yeah, through the kidneys probably, right? So if you have an impaired renal function, you're not excreting it appropriately, which means that your plasma levels are increasing, and you combine that with a drug that could have significantly changed her weight, and now you have a situation where effectively it's too much metformin, even though we didn't change the dosing.
this is basically what you just said. And-
Sorry?
No, you're fine. I did it to you all the time, so turn on fair play. You can see that renal insufficiency led to this metformin accumulation, and you end up with increase of. You cannot do gluconeogenesis, which is what we're going to talk about today. Talk about the twin towers of glucose, right? Glycolysis, right, which is what? Breakdown of glucose, and gluconeogenesis, which is the production of glucose, right? So we're talking about how we kind of balance those two things out. And when we talk about metformin, and actually metformin is kind of an interesting drug because it affects all along the metabolic pathway. So, and if you're a type 2 diabetic, or a lot of other diabetics, I suspect you're probably going to be on metformin. Even the pre-diabetic people take metformin. It's been around forever.
It's also used for polyovarian cystic disease and a couple of other conditions as well. So you'll see a lot of people on metformin.
We'll talk about it also because it affects, we'll talk about it again because it affects the electron transport chain too. So it affects multiple places. This is the structure of metformin. There's nothing overly exciting about that except for what? What do you know? What stands out like a sort of, like a, you know, what's the metaphor I'm looking for? My metaphors are getting mixed. Sticks out like a sore thumb. What sticks out like a sore thumb to you? Yeah, we've got a lot of amines there, right? What do we know about amines? What kind of interactions can we do with amines? We can do hydrogen bonds, right? We can also do sometimes electrostatic interactions, right? If the amine gets protonated, we can get electrostatic interactions going on there. So there's a lot of different things that can happen here. And you can just see without that many amines in there, 1, 2, 3, 4, 5, you're going to have all kinds of opportunities to interact. Just as a backdrop, so metformin comes in through hepatic cells and pumps in. It can block in the mitochondria complex 1, which this is in the electron transport chain. We'll talk about that in a few weeks. And then it also can interface with your mitochondrial glycerphosphate dehydrogenase, which is this MGPD right there. And then that can go and block gluconeogenesis in multiple different places. So we'll start talking about that. Gluconeogenesis is a pathway, and we'll talk about all the different places that we will see this occurring. All right. Backdrop. Metabolism is composed of breakdown, metabolism. We're going to break down fatty acids, monosaccharides, which are what? What are monosaccharides? Yep. What's that broad category called? Carbohydrates. And amino acids, right? Which is what? Proteins. So fatty acids, carbohydrates, and proteins. All these are broken down into smaller molecules, polymers, and we either excrete them or they can get recycled and put into anabolism, which is where we take and we add energy to a system, and then we actually make things with them. So some of the important thing is all of these are in pathways, right? So we have these long chemical pathways. What is the necessary ingredients in pathways? What do we have to do to build a pathway? Energy, enzymes. How do we regulate them? Do they just keep going? Yeah, there's feedback mechanisms, right? There's coenzymes, right? There's all kinds of different things that go into pathways. So right now, we're going to talk in a little bit. We're going to talk about all the different coenzymes that are important. What's our major energy source in all this? We've talked about it dozens of times, and hopefully you already have a pretty good handle on this. ATP. ATP is going to be our energy. It's going to be our currency for life. And basically, we're going to oxidize. We'll talk a little about oxidation reduction. Hopefully that doesn't spring back too many scarring memories from organic chemistry. That's in chemistry. That's the one area that one of the areas that comes back to haunt you from organic chemistry. I always tell people that you don't really need organic chemistry for this class, but that may be the one area this is one of the areas that it's a little bit helpful if you've had it. Metabolic pathways commonly occur in the cell, and in particular, a lot of things happen in the mitochondria. So we'll talk a little bit about mitochondria. These metabolic pathways are very, very highly regulated. And, Matt and I had quite a debate this week about drugs that affect this pathway, right? Or these pathways, I should say. And frankly, there's not a lot. Why do you guys think that would be? Why do you think that's true? It would be bad, right? If you don't have something that's working in these pathways, you're probably either you have high morbidity or you have high mortality. There's not a lot of I'm a little bit sick, right? It's pretty much you're massively impaired or you're dead. So those are the two outcomes that occur based on if these pathways don't function. A lot of the other pathways, you can have a little bit of, poor phenotypes, but frankly, the other ones, or these pathways, you just really can't. This is we're talking, so there's three, I'm going to skip ahead and then I'll come back to this real quick. We're talking about carbohydrate metabolism. This is our food intake, and then we're going to we're going to break our complex carbohydrates and our proteins and our triglycerides. They're all going to feed into the glucose, or through to glucose, and then we're going to go to energy production, which is our pathway we'll talk about tomorrow, which is the Krebs cycle and electron transport. But all of these kind of feed in at the end of glycolysis. Okay, we're going to primarily talk today about this pathway right through here, which is all complex carbohydrates. I think on Wednesday we talked about proteins. We may push that back to Monday of next week. What do you? No? Okay. She said no, so we won't do that. Let's talk about cellular respiration. This is a nice little
How do you turn that bite of food into a
I'm just going to send this to you. I forgot that I didn't realize I don't have.
You don't have speakers?
It's coming out of my normal speakers, not.
How do you turn that bite of food into a chemical that a cell can recognize and use as energy? The first step is altering the food into its component chemical compounds and then getting those.
Guess I should have checked that because it works just fine in Gate City. So I'll send that out later. All right, so we're going to start with complex carbohydrates, right? Glycogen, right, which is a major component in foods. Glycogen, as you might recall from last week, has branch points, right? So we have long chains of carbohydrates that are put together with alpha-1,4 glycosidic bonds. We have branching points where we have alpha-1,6 glycosidic bonds. What do we know about enzymes in relation to this? They're very specific, right? So can an enzyme that breaks down an alpha-1,4 glycosidic bond break down an alpha-1,6 glycosidic bond? No. And in fact, we don't even have very many enzymes that can break down glycosidic bonds in general. I actually don't know why. It'd be kind of nice to be able to eat trees if you were desperate, right? I mean, no one really wants to, but if that was the only thing you could eat, wouldn't it be nice to eat a tree? If that was the only, well, I assume you'd probably be able to you'd develop taste buds, right? We would evolve taste buds to like trees or like leaves or stuff, right? I mean, you guys think I'm crazy. That's okay. We don't have a lot of enzymes that break down because these sugar structures are everywhere. That's the major component of structures for all plants are these bonds here. There's different kinds of glycogen storage disease issues where we can't break down. You don't need to know this for the test. This is just informational. There's 4 different types of glycogen storage deficiency or storage disease issues. Type 1 through 4. They come in at different points in the pathways. So at different points in every pathway, there's an enzyme that causes problems, and you can have a disease deficiency associated with it. These are not common diseases. You're not going to have a lot of people that know about, polyglucosan body disease, APDBD. Most diseases you've probably never heard of in this list. They're just not that prevalent. You're talking prevalencies of 1 in 100,000. If I put up these diseases in my 460 or 260 classes where I have 400 people, and I asked if anyone know anyone with these diseases, I get maybe 1 or 2. And frankly, the probability of someone just accidentally hitting the wrong answer is higher than them actually knowing somebody who actually has these diseases. Occasionally we see someone who does know these diseases, but there are things like, glycogen storage type 3 is 1 out of every 360 or 3,600 people in the Faroe Islands, right? So you're talking about some, and there's like diseases that are associated that are like up in Ontario. There's a small, Amish community that has, or Jewish community or something like that, and that's where you'll see these kind of diseases, the genetic abnormality. Again, glycogen, as a reminder, is this long branched carbohydrate. This is what it's going to look like. It's predominantly in the liver and muscle cells. This is what your liver and your muscle cells use for energy. What we have here in glycogen metabolism is we have a glycogen phosphorylase, and it comes in a dimer. You can see the dimer here in the blue and the green, and then you have your carbohydrates and your cofactors, and this enzyme then will hydrolyze it and create a glucose 1-phosphate. Okay, remember that because we'll see glucose 1-phosphate again. This has to happen inside of a cell for it to be effective. So once we get that's what happens before we get into glycolysis. My piece of bread, right? It's going to break down all the bread into, that process is going to break all that glycogen into glucose. From glucose, we're going to go into glycolysis. Okay, we're going to make some ATP, and from there we're going to go into the Krebs cycle. And because we're biochemists and we like to confuse you, we have multiple names for all these processes. Okay, so you will need to know all the names because you will see all the names. And I wish I could just say, hey, just learn the fact that this Krebs cycle is known as the Krebs cycle, not the TCA cycle, the citric acid cycle, or all the other names that we call that cycle, right? And I used to think for a long time TCA was the citric acid cycle. That's not true. I don't even remember what it said, the tricarboxylic acid cycle or something like that. So we literally like to name this cycle a lot of different things. This cycle right here sits in the middle, and actually all of the other things we talked about, your proteins and your fatty acids, all enter in. So sometimes people will give this cycle first because all the other cycles feed into this. Okay, I like to go with glycolysis first, and then we'll talk about the Krebs cycle, and we go to oxidative phosphorylation, and then we come back and talk about the other components. But this Krebs cycle produces ATP and carbon dioxide. Then we go into oxidative phosphorylation, also called electron transport chain, and that's where we generate the majority of our ATP. We can also go into fermentation, right, which produces either alcohol or lactic acid, and we'll briefly touch on that today. I think we've talked about that before, right? So that's the big picture about where things, we'll get through this glycolysis today. Tomorrow we'll talk about the Krebs cycle and oxidative phosphorylation, I believe, right? Isn't that true? I usually do them in multiple days. But so how does this work? So glycolysis, we make 2 ATPs, and we come out of the process with pyruvate. So besides that, we also have NADH that's produced out of this that can go into electron transport. Produced straight up, the net is 2 ATP. Krebs cycle produces net 2 ATP. We also have electrons and NADH and FADH that then get transported to the electron transport chain. You'll notice that glycolysis happens in the cytoplasm. These are the two pathways happen in the mitochondria. And you'll notice the electron transport chain is the king of producing energy. It produces 32 ATP out of that cycle. We produce a lot of the intermediates, including the NADH and FADH, in these two other cycles that feed into this, but this is directly where, and we've talked about the FOF1 ATPase, and that's the powerhouse of electron transport. That's really where all the work is done. So this is the common metabolic pathway I talked about, and I'm just going to mention it because I already did, but I'm going to mention it again. So we come in with glucose, and it comes into this Krebs cycle. We can also, from other pathways, we have amino acids, and we also have our fatty acid cycles will come into this Krebs cycle, and this is called this is known as the common pathway for almost all of the power generation that we use. There are 4 important, and the reason we're going to talk about this right now is because there are 4 important coenzymes that or cofactors that are used. We'll start with ATP, and we'll go over in a little bit of detail on all of them. You should know these 4, especially. We'll talk a little bit about coenzyme Q when we talk about electron transport, but these are the 4 ones amongst the glycolysis. So we have ATP, that's obviously our storage for energy. We have NAD+, which is how we transfer electrons. It's an oxidizing agent, and that's how we transport electrons into the other pathways. We have FAD+, which is also another way for us to transport electrons. And then we have coenzyme A, and that transfers acetyl groups around. So, which we'll talk about why that's important when we talk about electron transport. So there's 4 enzymes you should know. We'll start with ATP. Most important compound because it's the spot at which we're going to, or the molecule by which we're going to transport energy around. And it is a nucleotide. Obviously we've got our adenine. We have our beta glycosidic bond that links our base with our sugar residue. In this situation happens to be an RNA molecule. We have a phosphoric ester here. So we have carbon, oxygen, phosphate with a double bonded oxygen, a phosphate right there. So that's a phosphoric ester. And then we have 2 phosphoric anhydride bonds. That's where our power is. So we break these bonds, pop them off. That's where we get the energy to do the work that needs to happen. Enzymes use it to facilitate the work that they do. So phosphate esters are thermodynamically unstable, but they're kinetically stable. So kinetics is what? Bingo, right? So it's how fast it occurs, right? Thermodynamics, whether the reaction is favorable. So it's thermodynamically unstable, but kinetically it's stable. Okay? And that means that we can use it. Enzymes can use that reaction by controlling the kinetics of that reaction. So because they're stable, because the negative charges, because all the negative charges on there, that resists hydrolysis. So until we start using them, in enzymes. So they're ideal regulatory molecules. And there's a couple different enzymes that are really important. We're talking about phosphate groups. You should know these because they're actually, a lot of them are drug targets. So when we add, does anyone know this? When we add a phosphate group, does anyone know the enzyme class? It's a kinase, okay? So those are very, very common drug targets, and you should probably just put that in the back of your head for reference. And then it's removed by phosphatases. So if we hydrolyze this phosphate, the hydrolysis of ATP to ADP, we generate 7.3 kilocalories of energy. If we go from ADP to AMP, we generate another 7.3. That's the common mechanism by which we will use energy. We go from ATP to ADP and ADP to AMP. Generally, we go ATP to ADP, but you'll see it both ways. That was a little joke from. Is that show again? No, it's not Mr. Mom. The one with Psych. It's from Psych. I've seen it both ways. Okay. Okay, so that's ATP. Next, we're talking about NAD plus and NADH. So, both of these things are oxidizing agents, right? NAD plus is an oxidizing agent. FAD plus is an oxidizing agent. And they're going to be reduced, which means electrons are going to go onto them, right? So they're an oxidizing agent. They're going to be reduced. And then that's going to be put into electron transport down the road. You following that? This is where you're going to have to go back to your I'll spend a couple slides on oxidation reduction reactions, because then in electron transport, they get oxidized again, right? All those electrons are stripped off, and they get put back into it and recycled. Again, and I like this structure right here. I don't know if you can see it very well, but it shows that this molecule is not just flat. There's a lot of intricacies in this molecule here, and it allows you to kind of give a visualization, at least in my book, about what's going on. All right, ATP is kind of, as far as energy goes, it's kind of in the middle, which makes it a good energy currency. So things like glucose and glycerol, right? It has much higher energy. So here's ATP as far as energy. So on this axis we have how much energy it's stored. So things like glucose and glycerol is low. ATP is kind of in the middle, and things like bisphosphoglycerate and phosphoenol pyruvate and creatine phosphate all have higher energies. That makes it a good currency. So I can transfer back and forth and move it around. Since it's in the middle, it can be used as fuel. Here are some of the standard free energies of hydrolysis of compounds that are phosphorylated. You can kind of see, again, ATP kind of fits right in the middle there. So, as far as how much ATP it actually it carries. How did that get in there? I must have had a brain cramp there. Anyway, okay, so let's get back to NAD, right? I must have pasted it in the wrong place when I was moving things around. Okay, so sorry, okay, let's get back to NAD. NAD plus. So what ends up happening is we have NAD plus, and we're going to reduce this by adding a proton and obviously electrons in there to get NADH. So that's the reaction you're going to see over and over and over in our glycolysis and other stuff. Actually, valine glycolysis. So you're going to see this a couple times in glycolysis, and then this NADH is going to be used down the road. So NAD plus is a cofactor in coenzyme, in the process, and that is going to be NAD plus is an oxidizing agent. It's reduced to NADH, and from there we're going to use it in the electron transport down the road. FAD is also going to be reduced. I always have to look at that and actually in my brain do it because it doesn't make sense to me. It's an oxidizing agent that's reduced. I always have to go back in whenever I do this. So this is FAD plus. We add 2 electrons basically to FADH, 2, and now it's going to be that's the reduced form here, and that's going to be added again into our electron transport chain, And from there, we're going to generate more energy with those electrons. All right. The last important coenzyme is coenzyme A, and you'll see this abbreviated CoA. You'll see that this is acetyl-CoA, right? You'll see this CoA here, very, very common. And it's actually a fairly large molecule, but it has an acetyl group in there. And that acetyl group is important because it often contains a unit of ADP. And it's also got a high energy thioester in there, which right there. So it's also important in that sense. And this kind of shows the full glory of coenzyme A. So we got a phosphorylated ATP, we got a pantothenic acid, and we got a mercaptoethylamine right there. All of these put together in this crazy structure right there with tons of different functional groups in there. And we're going to use that as, an acetyl and acetyl carrier, and for the thioester energy, as well as the ADP. All right. One glucose molecule. We start off in glycolysis. This is the kind of thing that's probably pretty important for you to know. A lot of I'm going to say this. Amanda and I've had this discussion with multiple professors. In theory, we should make you memorize all of these pathways and the enzymes and the inhibitors, and the cofactors, and the coenzymes, all that kind of crap. And I find zero value in that. But there's going to be some memorization you're going to have to do. And I'll just say right up here that this is the one. Okay? You need to know how much energy is produced out of each cycle, and what the final products are. When I take one glucose molecule, okay, you need to know this slide right here kind of gives you a really good handle on it. Glycolysis yields 2 ATPs, and then we also generate 2 NADHs and 2 FADH pluses. Those go to the electron transport chain, and that's going to give you 3 more ATP. Okay. Stage 2 is what? Where we convert pyruvate to acetyl-CoA. Krebs cycle, also known as TCA, citric acid cycle. Okay. We're going to generate 2 NADHs, okay, and for a total of 5 ATP. We're missing something there, but we'll get back to that. TCA cycle, that's this stage. Oh, sorry. This stage, this is the step in between between glycolysis and citric acid cycle. That generates 5 ATP. Citric acid cycle generates it generates 2 ATP, 15 ATP from NADHs, and 3 from FADHs. We're going to get a total of 30 ATP molecules out of this whole process, which is pretty substantial.
Do we need to know this just for the test, test, or also the final?
It's mainly, we're going to have it on this test, but it's really going to show up on the final. Yeah.
On the other slide, it says there's 32 ATP.
Yeah. Yes. Did I say on this slide? Or on this? Next slide? Or
It was back. It was on slide 15.
yeah, this happens all the time, right? So I'll go back and figure out where one of them is wrong, or what happens. So there's different things that can happen in these cycles, right? And this is not the first time of this. Sometimes there's things like GTP that's produced, so some people consider GTP not the same thing as ATP, and so those are the kind of things that happen, and I'll figure out what happened with that, and I'll tell you why. I bet that's what it is. I'm willing to bet that they consider the GTP in here, or there's something that's like that in this whole process that we missed.
Most people say it's like 28 to 32.
Yeah, there's some little, but the important part really is we're not going to give you an answer that's like 28 or 32. It's really going to be multiple choice. You're going to see like 3 and 300 and stuff like that. So you're not going to have to worry about whether, as long as we're in the ballpark, you're probably okay. And I would just know that this one produces 2, and this one produces 2, and then most of them are produced, right, in electron transport chain directly. So you'll notice all of these cofactors, right, are coenzymes. I keep saying that. So when I say cofactor, you just know I'm saying it wrong, but it's a coenzyme. But these coenzymes here are put down into electron transport, and that's really the important part of where a lot of the energy is produced. So what we end up needing to have is we need to have the amount of energy we're generating and the amount that we're utilizing need to converge, right? And that's the regulation mechanism by which we're going to be, we're running around, cells are running around doing their thing, and we need to have our energy meet and converge on a spot so that we are actually generating as much as we are using. And that's that regulates the metabolism. So we're also, here are the two activated carriers again in all their glory, and this is where the reactive sites are. So this is FADH. You can see that there's two activation sites right there, and this is NAD+, and this is the reactive site right there. So they get reduced and oxidized at those two sites.
Is one more reducing than
Yes.
the other? Which one?
You're testing me. NADH. NAD+ is not much more. I think it's like, I don't remember the exact numbers, but it's like 2 to 3 more than FAD+. And that happens in electron transport. I haven't studied that yet. That's tomorrow. I have to go back and refresh why that
You
is.
guys are going to want to be able to compare NADH and
See, she just sniped me right there.
oxidized.
Great snipe right there. Yes, so you'll need to know
I
that.
wrote the test today, so.
And I wrote that question last semester. That's but I don't remember. Well, yes. So, yes. It produces more energy. And that has to do with complex number 2 in the electron transport chain. But I need to go, when I give the lecture tomorrow, I'll explain why. I don't remember off the top of my head. All right, so here's the, so this is acetyl-CoA. It's an activated carrier. So here we have acyl-CoA and acetyl-CoA. You can see the only difference is acyl-CoA has an R group there, whereas acetyl-CoA really has, this is the acetyl group right there, and we can transport them. These oxygen esters are stabilized. They're resonance structures, as you often see with double bonds. And those are not available to thioesters, right? And so thioesters are a lot more unstable. So you can see that this structure does not form resonance, whereas this one will. So there's not the stability, and so this is a lot less stable. So that becomes an activated carrier of the acetyl group, which we'll talk about a little later when we get into the when we get into the actual glycolysis pathway. So here's some activated carriers in metabolism. You can see what they carry around. So really the idea is we're just going to be moving stuff around. Groups, electrons, right? So we got, ATP, the group that's moved around is phosphoryl. We've got all these electron groups. We've got these acyl, or acetyl group. Aldehyde, biotin is CO2, all the way down through nucleoside triphosphates. In this whole process, we're just moving. We're moving electrons around, and we're moving groups around to get energy. And those are activated carriers. Okay, last slide before the break. We have regulatory controls on everything. everything is regulated. Really, really important to know. We are regulated by three prominent things: the amount of enzyme that's present, the catalytic activity of the enzyme. What's that from? Was that 2 weeks ago? What's the catalytic activity of an enzyme? What variable name do we give that?
Is it KM?
She tricked you. she baited you.
So you have KM, you have KCAT, you have K1, you have K2.
You have Vmax.
What? It's KCAT.
That's also known as your turnover number, right? So you'll see that terminology used.
I remember that KCAT is the same as K2.
And we also have the accessibility of substrates. Now, in a normal environment, that would be associated with what? What's the main component of accessibility? Concentration? Right? Wouldn't you say that? If there's more concentration, that you have more accessible substrates, right? But we also have membranes, right? There's all that stuff that there's one advantage to this course, is like, when I talk about membranes, that was last week, not 3 months ago. So at least you have a frame of reference for what we're talking about. Because like if I was teaching this in 461, that would have been 3 months ago, right? From 460. So that was last week or 2 weeks ago. So all of these concepts, regulation of these pathways, go back to what we did a few weeks ago. All of them. The amount of enzyme present, obviously that's the enzyme chapter, catalytic activity, right? And the accessibility of substrates. That's a concentration issue. It's also a membrane issue, which we talked about last week. All of these things, we actually create membranes to adjust the accessibility of these substrates. So that kind of gives you an idea of where we're sitting with all this. I need a quick drink. I'm only a third of the way done, but my voice is not going to go all the way if I don't get a drink. So let's take a 2-minute break, and I'm going to get a drink, and then I'll be right back. We'll probably have 2 breaks today.
Pretty sure it's 1.5 for everything. Thank you, writing is 2.5. Yeah.
So the difference between NAD plus and FAD is NAD plus more reducing or more oxidizing.
And like Ben said, we'll cover that more in electron transport chain, but we just had them both up there, so I wanted you guys to start thinking about how we can.
Dead tea. Sure, if you want to. Just dump out the dead tea. Thanks. All right, I'm going to start back up. So now we're going to dive in. General idea out of glycolysis is taking glucose. We've got a 10-step process, not to be confused with a 12-step process or programs. And then that goes into pyruvate, and we can either do anaerobic lactate or aerobic, which is going to go to acetyl-CoA. That acetyl-CoA will then go into the citric acid cycle. And if we were yeast, we could ferment stuff, make alcohol. That'd be a third pathway, anaerobic pathway. This right here is the full process. So has all the structures. And I, for the test, I think I use this. Don't I? I think I actually put this.
Yeah.
I think we actually put this on there. So this is the citric acid cycle. Why do I have the citric acid cycle? This is the one I want. To go back for a minute. Huh?
Well, this is the actual diagram that's on the test. But all of your cycles will be on the test.
This will be actually for tomorrow's lecture. So when I copied over that, I must not have noticed that. I like that diagram a lot, so I probably just got enamored with it and put it in there. Okay, so this is the glycolysis cycle. We're going to start with glucose, okay, with hexokinase, which I think you've seen quite a bit of, and I actually will show a quick video of hexokinase here in a minute. There's some really good Pymol and other diagrams of hexokinase. This actually costs an ATP. So we're going to take glucose. Hexokinase is going to actually add a 6-phosphate. You might recall from the glycogen section that I just talked about that out of that we actually end up with the glucose 6-phosphate. So this first step only occurs if there's only just normal glucose. Second step is phosphoglucosamidase. Thank you. And that's going to take, it's an isomerase reaction, so we're going to take glucose 6-phosphate and we're going to make fructose 6-phosphate out of it. So that's what I was
So you're going to have to know them because some of the questions actually refer to the name of the enzyme. So you won't be necessarily plugging them into a pathway per se. Like we won't give you the blanks and you have to plug everything in. But you have to be familiar enough with the whole process to know that if I ask you what enzyme converts glucose to glucose 6-phosphate and I give you 4 of those enzymes, you know which one to choose.
This is not necessarily, again, as I mentioned, this is not my favorite thing to do to you, is make you memorize all these, because there's also the regulatory. This is not the regular, there's diagrams that have all the regulatory elements on these things too, and have all the cofactors on them, and coenzymes. And so from here, I don't have a good answer for that. So I would note the stuff on your study guide. So if it asks you on the study guide to memorize it, that's what you need to know. Fructose 6-phosphate. We're gonna add another ATP. So this is another phosphofructokinase. So it's gonna cost us another ATP. So we've now cost ourselves 2 ATP. But we end up with a fructose 1,6-bisphosphate. Okay. From there, we end up with the aldolase is going to cut this basically into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Okay. From there, we're going to use triose phosphate dehydrogenase, and we're going to end up with 1,3-bisphosphoglycerine. So there's an isomerase reaction that occurs here to convert that the molecules, and then we end up using triose phosphate dehydrogenase, and this is where we're going to start making our NADH. So we're going to oxidize, we're going to reduce, NAD plus is a reducing, or an oxidizing agent. It's going to be reduced to NADH, and we end up with 1,3-bis-phosphoglycerate, BPG, and we end up with 2 NADHs out of this.
So do they both, where do these come from? Do they each come from the different?
Fructose 1,6-bisphosphate is cut basically in half. So we have a C6 here, and now we end up with a C3, and we end up with a C3. They're isomers of each other, and so the isomerase converts them both to dihydroxyacetone phosphate.
No, the other way.
Thank you. Glyceride 3-aldehyde. Glyceraldehyde 3-phosphate. Thank you.
Because see, the pathway only proceeds through glyceraldehyde 3-phosphate.
Oh, that's true.
It doesn't proceed through dihydroxyacetone phosphate.
Thank you.
And in fact, it's kind of interesting because the dihydroxyacetone phosphate, if you have enough of that, that will shunt off into a different pathway, and it goes someplace else. So the other thing to point out is if you look at the end of each one of those enzymes, it tells you what classification of enzyme you're looking at. So when you see the kinase ones, know that you're going to use an ATP because you need a phosphate group, because you're going to add a phosphate group. When you say an isomerase, know that you've got 2 isomers, and you're going to convert between the 2 isomers. So if you can't remember exactly what the enzymes are doing, but you only remember the names, in theory, you can kind of place them where they belong.
So that's step 6. We're going to go a little bit on detail on some of these first little steps, then we'll go back and finish up the process. So this gives you an idea of what. So we're going to take a normal glucose. We're going to take ATP, the enzyme is hexokinase, and we're going to add this phosphate group up to this number 6 carbon. It's a glucose 6-phosphate up there.
One more thing.
Sure.
Remember that hexokinase, that reaction to go to glucose 6-phosphate is highly favored. In fact, it's considered kind of one of those regulatory points where you can't go backward to glucose. So it's highly favored to go to glucose 6-phosphate.
And there's a couple different cofactors. There's a lot of magnesium in here, so and manganese. So there's a divalent metal ion that's required for these. That's going to allow that enzyme to work. Now this is I love this diagram. I think this is a really good. This looks like an enzyme, right? If you've been following, you can see here's the glucose molecule on there. Here's the ATP and ADP site right there, and in this enzyme actually folds in and produces. And I'll show it to you here as well. In the animated form. So you can see wherever glucose is going to come in there, the active site. It's going to go from a T to an R, and then it's going to produce the reaction that occurs in there is going to be glucose 6-phosphate to ATP. Glucose 6-phosphate. And that's what's produced out of the process.
Do you guys understand T and R states? We haven't really talked about that, but we keep using that term. Just remember, it's basically your active and inactive states.
Tense and relaxed. Step 2, it's a conversion to fructose. It's an isomerization reaction. Basically, we're going to go from an aldehyde to a ketone. So glucose here is in the open chain form, and we're basically going to then move it to ketone in the fructose form. So, and then it's circularized, and this is the final form. So this is the isomerase that goes from glucose 6-phosphate, okay, and we're going to make it into fructose 6-phosphate. And the reason this occurs is because glucose 6-phosphate is not easily cleaved into 2 3-carbon chunks. I think evolution is crazy. The fact that we have all these like 10-step processes and there's dozens, if not more of them, and we do make energy out of this is just crazy to me. So from here, we're going to use phosphofructokinase. And we're going to add another ATP. So we're going to end up, this is basically going to make the molecule equal. So when we chop it in half, we have 2 of the equivalent amount of carbons and 1 high energy phosphate bond there. So we're going to go fructose 6-phosphate, phosphofructokinase will add an additional phosphate on. So it adds this phosphate on. Now you can see we're balanced. So we have a 6-carbon, right? And we have a phosphate here and a phosphate here. And we're going to end up with a proton that's produced there that we'll later use.
The other cool part about that molecule is fructose 6-phosphate can pass in and out. Fructose 1,6-bisphosphate gets trapped, and you can't go backward. So it's the way that they trap, the way that the system traps fructose.
Oh yes, an important note is this is an irreversible reaction.
Irreversible reaction.
And we'll talk about this again when we talk about gluconeogenesis because that's also happens in that pathway. Next, again, we're going to cleave. I got ahead of myself when I started going on that pathway. But whatever. This is where we cleave into 2 3-carbon species at the beginning of the second stage. So we're going to take our fructose 1,6-bisphosphate, and aldolase is going to make our dihydroxyacetone phosphate and our glyceraldehyde 3-phosphate. So again, dihydroxyacetone can't be used, so an isomerase is going to change that. Triose phosphate isomerase is going to make it into glyceraldehyde 3-phosphate. This is basically going to isomerize dihydroxyacetone phosphate.
So if the conversion from fructose 6-phosphate is just not if it's an irreversible reaction, why don't they just hold the arrow heading to fructose 1,6-phosphate?
So this one is irreversible. And you notice that this one is reversible. Did I say it was irreversible?
No, but if you go back to the previous slide, I think what Jacob is referring to is you have that tiny little arrow indicating that it could potentially go back, but for all intents and purposes, it's irreversible. So they put, it's kind of one of those, let's hedge our bets, it's a CYA thing, right? Let's hedge our bets because we always say, well, that's never going to happen, and then we find a way that it does. So effectively, it's an irreversible reaction, but they always put just that little hint that, well, maybe there's something we don't know out there. That's why they do it.
So we have an oxidation reduction reaction that goes when we make from glyceraldehyde 3-phosphate. And this is one of the oxidation steps. So we're going to, this oxidizing agent NAD plus is going to be reduced to NADH. Our glyceraldehyde 3-phosphate is going to be, so we're going to add a phosphate group to it, and it's going to be oxidized at NADH. So we always have something that's oxidized and something that's reduced. And you should be able to identify what those are. Now, on the test, on our test, I think it says, I think we just say, is this an oxidation reaction, a condensation reaction, a hydrolysis reaction? Yep. So if it's a reduction, we're talking about the product and not NADH, not the coenzymes. So we will, and you can just reference that. Any oxidation, obviously any oxidation reduction, something has to be oxidized, something has to be reduced. So we don't say it's an oxidation and a reduction. We won't put those two answers in. It'll just be an oxidation. Does that make sense? So, whereas some of these are condensation reactions, they produce water. Some of them are hydrolysis reactions that they use water. This is an oxidation reaction. So the intermediate It has a large delta G, double dagger. We have a thiol ester intermediate that kind of reduces that, and that's that thiol ester linkage is going to be what's going to be oxidized. That's what the dehydrogenase is going to do. So it's going to be a coupled reaction. So the reaction is going to be coupled through a thiol ester intermediate. All right, so this is where our first ATP is produced. We're going to go from 1 bisphosphoglycerate. We're going to take ADP and 1 proton. Phosphoglycerate kinase. Notice the kinase is going to add a phosphate group to our ADP, and we're going to end up with 3 phosphoglycerate and ATP out of this reaction. Now, there's actually 2 ATP that are produced, right? Because there's 2 of them, because we had C6, now we have 2 C3s. So actually out of this step, we actually produce 2 ATPs. Out of this reaction, we produce 1, but there's a total of 2 because we went from glucose, which is a C6, to 1 bis phosphoglycerate, which is actually, there should be 2 of those based on 1 glucose molecule. From here, we go from 3 phosphoglycerate through phosphoglycerate mutase to, and basically we're going to just move the phosphate group to 2 phosphoglycerate because that's energetically favorable. This is a condensation reaction because water is a product of that reaction. You'll want to know, if you see water coming out, you'll want to know that that's a condensation reaction. So that an enolase is going to produce that, and we end up with phosphoenol pyruvate. And basically what we're going to do is we're going to create a double bond right there. From here, we're going to produce another ATP, which actually produces how many ATP? 2. So we've used 2. We created 2 in the previous step, and now we're creating another 2. And you might see these as GTPs. That's very common in the discussion. So now we've created another ATP pyruvate kinase by cleaving off this phosphate group. This step here is just because this intermediate is less stable and allows us to go to the pyruvate step, which is the end of glycolysis. Here's what you see. Here's the, that face right there is exactly how I feel about that chart, right? Because this is the whole enchilada up there for you to love. Tells you the reaction, the enzyme, the reaction type, how much energy is used or generated out of it.
Are we supposed to know the energy too?
No. If you study the study guide, you'll do fine for this test. We'll have another discussion week of finals. We'll go over some examples for the final. I think the last Wednesday I'll do a study, a review session for the final. So I don't know, something in there. It's fun, but it may be more valuable to do. No. I take, so when I write the tests,
Enforcement guard secret.
you have to be part of the secret society. I had to sign over
Or
my
second.
my second born and my social security number, so they would ravage my credit if I gave out the information. No, it's pretty serious. I'm talking about, we can't order the test. We had to have the dean or the department chair of chemistry run the test. They
We deliver them in a sealed packet. Sign for them.
right. We have practice. We have professional copies. So what we do is when we write every week's exam, I sit down and I look at the type of questions that you're going to see on the final, and I incorporate those into the test. Those types of questions.
Do we have all of the previous tests?
You will have seen a similar question on every that you see on the tests. You will have seen it in the unit exams
Or in the study guide.
or the study guides. And I'll go over the study guides and give you an idea of what you should focus on. And I will tell you this week is one of the more heavily.
It's very heavily weighted between this week and next.
And they like enzymes.
We like kinetics a lot.
They like kinetics. All the fun stuff that you guys like, they don't like. Like DNA stuff, they're like, whatever.
I think there's maybe one DNA question.
Protein is very heavy on the test. So
So you've already seen the test?
not sure which that was in. I only have professional
Oh, it's
copies.
the same test.
We might give you the same test. I think we have to look and see what the.
Let's see what the rules say.
Don't get any ideas about breaking into our office either.
I have spider security,
All right, so so we just went through glycolysis, right? So let's just take a big step back and kind of regroup and see where we are. So, glycolysis, we go from glucose to pyruvate, okay? We generate 2 NADHs, and we're going to produce 2 ATPs. That's glycolysis. We're going to go into cellular respiration, which includes, just to confuse you a little more, because we include, if I'm going to talk about citric acid cycle, electron transport, or oxidative phosphorylation, cellular respiration includes TCA and oxidative phosphorylation. So if I say cellular respiration, that actually means something in the grand scheme of things. It actually means TCA cycle and the electron transport chain. So we're going to cover this part today. Tomorrow we'll cover the rest of this. Today we talked about all the cofactors that or coenzymes that exist, and glycolysis and gluconeogenesis. This is kind of an informative chart, just if you're curious and you need a quick lookup for, it has a lot of good information here. That was a stage, so glycolysis, Krebs cycle, and this is anaerobic respiration, so this is if you're going to go into a lactic acid cycle, or if you're gonna ferment, I think it'd be fun to do. I think we should be able to digest plants, and I think we should be able to ferment. I think you could self-medicate then, right? I meant like all plants, right? Like we can eat starches, right? But we really, and I don't know why. Look, I don't really have it. I don't even like kale. I don't even know why I say that I want to eat trees. But I think it'd be, I don't know why I can't eat trees. It's one of those questions that keeps me up at night. I mean, it's sugar, right? Is it not sugar? Right? It's not the kind that we like, but it's made of sugar. Why can't we eat it? Okay, sorry. I just find that a very fascinating question about why we didn't evolve. You'd think that at some point along the line they'd be like, and like wood doesn't digest very. I mean, there's not very much things that break down wood. Why is that? It must be thermodynamically unfavorable. Obviously, I need to look that up. But it's got to be thermodynamically related, right? Because we'd find some way of dealing with that otherwise, right? Wouldn't you think? There'd be some other kind of organism that came in that existed.
I bet it acts as a carrier. Because you can't break it down, it probably acts as a carrier because of the fiber that allows your digestive system to work better.
Anyway, sorry, I didn't mean to get you off on. No, I didn't. I just don't know. I mean, I don't know that we could ever find that out. I'm just
We'll have the discussion.
That'll be the first question I ask God when I die. Why can we not eat trees? Okay. Amount of ATP produced, glycolysis 2, 2, Krebs cycle 2, electron transport 32.
Here they say 32.
I'm gonna have to go look that up and see why. I don't know the answer to that question.
I don't remember.
Anaerobic respiration is much less efficient. We get 2 ATP versus 32. So you can see that we need glucose. Here are all the coenzymes that we talked about. And here in anaerobic respiration, we need pyruvate and NADH. And here are the products that we're going to get out of this. So here we're going to get pyruvate, ATP, NADH. Actually, it's pyruvic acid. 2 NADHs, which go to electron transport. Krebs, we end up with carbon dioxide, NADH, which we excrete, NADH, FADH2, ATP, and then we go into electron transport. Here we end up with lactic acid and NAD+, and in alcohol we end up with ethyl alcohol, carbon dioxide, and NAD+. Just as a reminder, I think there'll be 1 or 2 questions on this. Here's the oxidation reduction discussion. So we have an electron donor here. It's going to take, if we're going to, it's going to be the reducing agent. The electrons are then going to go to the electron acceptor over here, right? And that's a reduction reaction. This is going to be oxidized, right? So this is the oxidized donor, right? Here's the electron acceptor. That's the oxidizing agent. This is the reducing agent. We love to confuse people with this, so, but that's how a redox reaction occurs. So you can see, so this is lactate dehydrogenase. NAD plus is the oxidizing agent. It's going to be reduced to NADH, right? The product, lactate, is going to be oxidized to pyruvate. So we have one that's reduced, one that's oxidized, and we're off on our merry way. NAD plus, again, gets reduced, right? So we add electrons here, and so you can see that this nitrogen right here, right, NADH, oh sorry, right here, this NADH right here is where we add the electrons in the form of a proton and electrons. So phosphofructokinase is the most important control site. So if we have high levels of ATP, it's going to allosterically inhibit the enzyme. And you can look this up on PyMol if you're interested in this. So binding of ATP lowers the affinity for fructose 6-phosphate. So when there's low ATP, you can see that the reaction rate is higher, and when it's the velocity, which is also, that's your Km, and this is going to be high if, so this is going to be lower if there's high ATP. AMP competes with ATP but does not inhibit. Decreased pH inhibits the activity, so you'll see that in certain situations. Hexokinase is inhibited by the product glucose 6-phosphate. So if we're going to inhibit phosphofructokinase, that's going to lead to the inhibition of hexokinase. Phosphofructokinase is the first committed step of glycolysis. So glucose 6-phosphate can be converted to glycogen too. So we'll see this in a little bit. It can also go down to the pentose phosphate shunt, which is another pathway. It can go into glycolysis, or it can go into glycogen and storage. So glucose 6-phosphate has different options at the point at which it can go down. We'll talk about this pathway, I think, on Wednesday. Obviously, we talked about glycolysis today, and we'll talk about gluconeogenesis, where we create glucose and glycogen later. Here's kind of an additional regulation. So pyruvate kinase, if we have high blood glucose level, right, we're going to go down this pathway, right? If it's low blood glucose level, we're actually going to go this way, so we'll go back. So at the point that this is how we regulate this entire pathway, and this will show up when we do gluconeogenesis. You'll see this pyruvate kinase can go either way, and most of them in the pathway can. This gives us flexibility, and they're just allosterically changed, or concentration-wise, they'll go the other way based on what the situation is. So in the muscle, we commonly have glycolysis being, right? So we go glycolysis, pyruvate, and commonly lactate because we're burning so much energy. So that's active skeletal muscle. That can go into lactate, and it's going to go into the liver then. So, and then gluconeogenesis occurs in the liver, and then we go from there. We're going to go to pyruvate. We're going to go from lactate to pyruvate to glucose, and that glucose then can be sent back to your active muscle cells. So we're going to start going into now gluconeogenesis, which is the next part of the lecture, which is part 3 of the lecture. So I'm going to give 3 minutes here so I can get a drink. And you guys can go to the bathroom if you need to again, because I am not going to be doing very good if I don't. So I'll just say that glycolysis is probably twice as important for you to know as gluconeogenesis. So when you're studying for the exam, I would know glycolysis more thoroughly than, or if you had to prioritize, you will see glycolysis more than you see gluconeogenesis. So, gluconeogenesis is just the opposite of glycolysis. There are 3 irreversible steps, and in gluconeogenesis, there's a corresponding 3 enzymes that will get you over those steps. So here's glycolysis, right? Everything's reversible except for hexokinase, phosphoglucokinase, and pyruvate kinase, and all of these are going to be the ones that are not going to allow you to proceed. All of the gluconeogenesis enzymes are in red. The glycolysis enzymes in blue. And I'll explain the differences here in a little bit, but there's only 3 steps that are. I think I actually show this a little later too. Ah, here we go. Yeah, so there's 3 irreversible ones. That gives you a little bit better or different look at it. So there's 11 enzyme catalyzed reactions here. It can happen in the mitochondria or the cytoplasm. This kind of gives you So glycolysis is going to use ATP. It's going to generate ATP to ADP. Here we're going to add a phosphate, or take a phosphate off. So you're going to end up with a free phosphate group. Here are again, those three reactions that I mentioned earlier that are not reversible. At this step, the second step, and the second step. Gluconeogenesis occurs when you're in fasting, starvation, or on low-carb diets, or you're in intense exercises. So you start fasting for a couple days, or have low-carb diet for a couple days, and you're going to start dipping into gluconeogenesis. This kind of gives you an idea of how that works. You can generate glucose from lactate, glycerol, or glucogenic amino acids. Which we'll talk about when we talk about amino acid catabolism. So here we convert glycerol into through glycerol kinase, right? We're going to create glycerol phosphate dehydrogenase, and then we're going to go to dihydroxyacetone phosphate. And from there, we can go with the rest of the pathway. We can also convert CO2 to carbohydrates. This actually is photosynthesis, right? That's another way we can go. But no one really cares about that, right? You might see one test question on that, so if you're... Actually, I wouldn't waste your time. You're gonna have one question you're just gonna miss on the final, because unless you remember it from high school or somewhere else, because I don't really care about fun. Okay. Gluconeogenesis is the synthesis of glucose again from a non-carbohydrate source. It's not the exact reversal, but it's pretty close. So here's what ends up happening. I like this chart the best. So glycolysis, gluconeogenesis. And you can see that most of the enzymes are the same, with the exception of the first, the second, and the seventh step, which we'll show you on this chart. This is the equivalent chart. Shows you all the gluconeogenesis steps. Right, so here are the three irreversible steps. So we're going to go from lactate, citric acid intermediate, or glucogenic amino acids. It's going to come into through pyruvate into this step. We're going to go to oxaloacetate, phosphoenol pyruvate. Then we've got our last 5 steps that are similar to the glycerol or the glycolysis steps. Then we're going to go down these next all the way to glucose. It's basically reverse with the exception of the marked spots, which are, again, identified here. So instead of hexokinase, we have glucose 6-phosphatase instead of phosphorylated kinase. This is fructose 1,6-bisphosphate, and down here instead of pyruvate kinase, we have PEP carboxykinase and pyruvate carboxylase that allow us to go the reverse step. So we can convert glucose to other hexoses. Why do we need other hexoses? Yeah. Well, yes. What do we need other 6 carbons for? Yeah. No. That's the right answer. What else do we need 6 carbon carbohydrates for? I think we said it last week, right? What do we use carbohydrates for besides energy? Yep. How about all our post-translational modifications? Yeah, linkers, all kinds of other things. So glucose is converted to other hexoses and other polysaccharides, and we use UTP to form a UDP intermediate, and from there we can then convert those molecules. We can also do glycogenesis, which is the synthesis of glycogen from glucose, and that allows us to, glycogen is large branched chains of glucose, and so this is basically how that occurs through the UTP pathway. Okay, so we talked about metformin earlier. Right now I want to do an activity where you find and identify the mechanism of action. I'm going to give you 5 minutes to do that, to see if you can figure out, look it up online, figure out if you can figure out how this works. And we're going to have a discussion about it. Bodies. Right, so does anyone want to tell me what the mechanism of action for metformin is? What'd you guys find? Where'd you look?
The FDA says it decreases hepatic glucose production, decreases intestinal absorption of glucose, and improves insulin sensitivity.
Yes. So those are the three mechanisms of action. But guess what? I'm a biochemist. So that's like... Right, so what do I want? Okay, so where does that occur? Okay, so we talked about this earlier, right? So what enzyme does metformin block? What is MGPD? Okay, yep. So, act as more part of gluconeogenesis, right?
So it stops the production of glucose, right?
It does. It does. So where's glycerol phosphate dehydrogenase? Which step? So for gluconeogenesis goes, right? Pyruvate all the way through. We'll talk about how it gets to oxaloacetate here in a little bit. But it blocks it there, and blocks fructose 1,6-bisphosphate. Right? So let's go back down and see if we can find glycerol phosphate dehydrogenase in gluconeogenesis. Right there. Right, and if we go back and we look at the other, should have copied this one in. Down here. So what? So we've got multiple steps along here that it's going to block, as well as right in that FPBase. So that's an adenylate cyclase. So what websites did you find actually the mechanism of action? And if I wanted to do, let's say I wanted to do metformin as my drug, what would you start looking for in the video? How would you do that?
You just look up what it binds to.
That'd be a good start, right? What else could you do? What if you typed in metformin PEB? You might find what it binds to. I have a feeling it binds to a lot of things just because of the nature of what it is. But if I was making a video, this type of graphic right here would be one of my starting places for my discussion of my video, right? And then I would get into showing the molecular interactions of the molecule with MGPD and potentially with complex 1. This would be a good, solid effort if you just put this in. Probably not get you the full credit. Obviously, we're doing all or nothing. So you're going to want to show at the molecular level how that drug sits in that site. And you may not have a perfect idea of where that is, but you could probably piece it together by saying something like, "Oh, it's a competitive inhibitor of phosphoenolpyruvate," or sorry, that's a bad one, oxaloacetate, and this is the site that it binds to. We may not have the crystal structure of that molecule, but you can just see how this works. Or you could show this OCT1 and how the metformin is introduced into the cell, because obviously, right, it's got to get through the membrane.
So you said that doesn't work on crystal structures, you don't always have to use Thymol?
No, what I'm saying is you're probably not going to have the crystal structures together. So you'll have a crystal, I promise you, they'll have most of these as crystal structures, if not all of them. I haven't checked that, but I'd be stunned. I'd be stunned if it didn't. They didn't. And then I'd identify, well, okay, phosphofructokinase or whatever is inhibiting that, this MGPD and the whatever, and I would go pull up the crystal structure of that, identify the active site of the enzyme, and then just say, this is where it fits. You don't have to show the molecule there. You can just show, hey, these are the residues that interacts with it, and this is how it's hypothesized it binds in there. That makes sense? Does that answer your question? Okay. Oops, getting ahead of myself, looks like. All right. So the first couple steps, when we go from pyruvate to glucose, the first couple steps occur in the mitochondria. It actually uses 1 ATP. It requires biotin. Right? Are you guys familiar with biotin? I think it's a supplement. I think people think it's a supplement, right? We use it all the time in biochemistry because it binds, I think we even talked about this, avidin, right? It binds avidin really tightly. And so, this is the important biological purpose of biotin, because it's a prosthetic group carrier of activated carbon dioxide, and biotin is not carboxylated unless acetyl-CoA is bound to the enzyme. And so basically we're going to go from pyruvate to oxaloacetate in the mitochondria through this reaction under these conditions, when we have a carboxylated biotin and the acetyl-CoA is bound to the enzyme. So that's going to occur under your fasting conditions, and that's how you're going to get to this step right there. And you're going to burn up an ATP in that step. Pyruvate carboxylase, this is a tetramer, 4 identical subunits. This is going to carboxylate that. This is the enzyme that's going to catalyze that last reaction from pyruvate to oxaloacetate. And you're going to have a biotin in there. You're going to have a tetramer of this pyruvate carboxylase, and that's going to convert that. Phosphoenolpyruvate carboxykinase. So this is the next step that's going to occur in the process. So we're going to go from oxaloacetate to phosphoenolpyruvate through this phosphoenolpyruvate carboxylase, and that's going to it's going to be an endergonic reaction. That's the next step. This process occurs, it's pretty ubiquitous in plants, animals, fungi, bacteria. Almost everything's going to do this step. It's in humans, it's mainly going to occur in the liver, and to a less extent in the kidneys. Mitochondria is where most of it's going to occur, except for the cytosol, which is the last few steps of that process. So the first few steps are done in the mitochondria, the last are done in the cytosol. If we're in lactate phase, right, which is what happens when does this happen? Yep, in anaerobic conditions, if we want to be precise about it. So this is the Cori cycle. There is, I believe, on one of the exams, there is a Cori cycle question. When we get to the review date, I'll verify that. But the Cori cycle involves synthesis of glucose from lactate. So for generating a ton of lactate, we can go, we're generating a lot of lactate in the muscles. In the liver, we can go from lactate through gluconeogenesis and generate glucose, and then from there we can generate glycogen. So this kind of gives another example. We can also incorporate amino acids if we need to. So if we need to generate, this is the next step in the cycle, generation of fructose 6-phosphate. This is, again, irreversible, and it's unique to gluconeogenesis. We're going to go from fructose 1,6-bisphosphate through fructose 1,6-bisphosphatase. We're going to chop off one of the phosphate groups, and we're going to end up with fructose 6-phosphate. This is irreversible, and it's unique to gluconeogenesis. So this is step 2. So if we have fructose 6-phosphate, again, this is easily converted to glucose 6-phosphate, which can be converted then to glycogen. It can be done in most tissues, and it can also be easily transported into the lumen and into the cytosol. So if ATP is required, which is most of the time, we'll use glucose or glycolysis. If glucose is required, we can make gluconeogenesis, and this kind of gives an idea of the important, unique steps in the process. But that's kind of the energy balance that we have. There's reciprocal regulation, meaning if one's going, one's not going, right? So that's the important thing. The key is fructose 2,6-bisphosphate. So that's the activator of phosphofructokinase, or PFK. It's an inhibitor of fructose 1,6-bisphosphate. If your glucose level is low, it can't bind PFK, and that's how it's regulated. So the important part of this slide is they're jointly regulated. So if one's activated, one is not deactivated. If one's deactivated, the other one's activated. They're reciprocal. There's never like a stasis moment where they're both deactivated or both activated. One's going, the other one's, and that's because they have irreversible steps at certain points, and they also have enzymes that are, several that are reversible as well. So this occurs, ironically, it can occur with the regulation, with the phosphorylation of a single serine residue. So on the enzyme. So that it's an enzyme is dephosphorylated, and that's going to change how that works. So on a serine residue, this enzyme, it's phosphorylated, it goes one way. If it's dephosphorylated, it goes the other way. And that can occur with gene regulation. Glycogenesis responds to hormonal control, so you can have epinephrine or adrenaline that will activate this whole process, and that activates glycogen phosphorylase and inhibits glycogen synthase, and that's going to activate the adenylate cyclase and form cyclic AMP from ATP. That's going to regulate things. As I mentioned earlier, this is we're kind of on the tail end of things here. The reactions of pyruvate. Pyruvate's the last step. That's in glycolysis. Again, there's three options that we have. We can go under normal conditions to acetyl-CoA in the citric acid cycle, which is the topic for tomorrow. We can go into lactate, which is under anaerobic conditions, or we can, in yeast, they can ferment and go to ethanol. So lactate, if we had lactate dehydrogenase, or since we have lactate dehydrogenase, we can, this is what's used in the muscle. It's in a, lactate dehydrogenase is a tetramer. The interesting thing about lactate dehydrogenase is, and this is one of those things that I think is really fascinating about biology, there's actually two different types. It's a tetramer, lactate dehydrogenases, and there's two different types in the subunits. They can all interact, and different muscles or different, I should say, cell types and different tissues have different concentrations of each subunit. So there's an H subunit that's very common in the heart. There's an M subunit that's very common in muscles. So in the heart, there's 4 H's, and in muscle, there's 4 M's. And then there's a whole bunch of tissues that have a combination. Some have 2 of each, some have 3 of one and 1 of the other. And they have all different kinds of things. And that's for the optimal way at which you're making, allows you to make lactate from pyruvate and use energy. So when we make pyruvate to ethanol, pyruvate is made into acetaldehyde by pyruvate decarboxylase, and then acetaldehyde is made into ethanol using alcohol dehydrogenase. Pretty simple, straightforward process. And we also have the product of making NAD+. So if you want to know how to make beer, that's it right there. That's your formula. When we were at grad students, it was pretty common for Budweiser to try to recruit some of our yeast people, because if you want to be a brewmaster at a major pub, you're going to know all the biochemistry about, not your hobby brew guys, but because if Budweiser's yeast goes down, that's a big deal, right? So here's the first step in that process. The last thing we're going to talk about today is pyruvate, or the pentose phosphate shunt, also called the phosphogluconate pathway, or the hexose monophosphate shunt, because we want to make it difficult. So it's parallel to glycolysis, but basically we're going to be making our ribose 5-phosphate. Where do we need ribose 5-phosphate? Yep, for our nucleotides, right? So this pentose phosphate shunt, pentose phosphate pathway, phosphogluconate pathway is where it's parallel to glycolysis. And we're going to use it to, in this part right here, we're going to use it to make, there's several enzymes that occur in there, and we're going to be using it to make, and we'll talk a little bit more about this next week, but I wanted to introduce the concept today. So, and we're going to end with that right there and go back to metformin. The important part of this is, this looks like a hot mess, doesn't it? And I think it's important to understand that when we drug something, the one thing I want you to get out of this discussion is when we drug something, you can see how easily it gets to have serious consequences. Right? This is where you start to see side effects. This is why side effects exist, because you can see that when metformin comes in, complex 1 affects all those other different systems and all those different pathways, and you can easily start going into problems. And this is where drug interactions can even get more problematic, because you stop one thing and stop 2 different pathways, and all of a sudden you've got yourself all kinds of crazy crap going on that that you didn't realize what was going on. And some of these molecules that we're talking about will bind to 2 or 3 different enzymes because they're just very generic. So that's the important part of that lecture, or that slide right there. Do you guys have any questions on glycolysis? Was that enough for today? I suppose it's that way every day, huh?